Good morning, class. Today we will start a new lecture. It will be called the second lecture of this chapter 7 called Cell Cycle. And today we will learn about the mitosis division of the cell cycle. As I discussed yesterday, that cell division is a kind of a process in which we multiply the cell. First of all, it starts with an interphase, and in interphase there are certain substages. The first substage is called the G1 or called the gap 1. If the preparation or if the things that are to be made in the G1 phase, if they are complete, so the cell or the cycle will enter in the following stage, and that will be called as the S phase or synthesis phase. If it is not valid, or if it is not preparing some of its molecule, then the cycle or the whole cell will enter in the G0, which is called the resting phase. In S phase, we saw yesterday that DNA is replicated in this phase, and in the G2 phase, actually in the G2, the cell also prepare itself for the next division or the next event, which we people are calling as the mitosis. If we recall the concept of mitosis, which we have discussed in classes and in my lectures as well, that our cell is carrying the chromosomes, and the chromosomes, they are in the form of the threads. So in this process, first of all, we will discuss in how many stages it is divided. So we discussed yesterday that the first stage of the mitosis phase is called the prophase. This is called the first substage of the mitosis. So in the mitosis, if we look into the deep in the cell, so before discussing the prophase, let's recall the concept, or let's recall the shape of the cell of any plant or of any eukaryotic living organism. So this is called the cell membrane. This is called nuclear membrane. Inside a nucleus, we have discussed a lot of times, okay, there are certain things. The very first thing and the most important is its chromatin network. Then there is a central body called the nucleolus, and the fluid of the nucleus is called the nucleoplasm. We have discussed in the previous classes as well that in a cell, or if the cell is not dividing, so you will not be able to see the word chromosome, but you will find a dense network of the fine threads, and this network is called the chromatin network, which is actually the network of the threads called chromosomes. But as the cell is not in its dividing phase, so we will not be able to see any kind of chromosomes. So let's begin with the prophase. uh, you will see certain kinds of changes in the nucleus, as we discussed yesterday as well, कि जो हमारे पास सबसे पहले चेंजेस आते हैं या जो सबसे पहले माइटोसिस में जो डिवीज़न होती है वो पहले एक न्यूक्लियस की डिवीज़न होती है. इफ़ यू आर सेइंग द डिवीज़न ऑफ न्यूक्लियस, तो वी आर स्ट्रिक्टली मीन्स कि we have replicated here the chromosomes already in our interphase in the S phase. Now we will first of all divide this single nuclei into two daughter nuclei in the cell. Once the two daughter nuclei they are formed, then we will separate, or then the cell will separate itself into two daughter cell. तो as I discussed yesterday कि जो सबसे पहले होने वाला काम है उसको हम कैरियोकिनेसिस कहते हैं, जिससे जो है हमारा ये एक दिखने वाला न्यूक्लाई यहाँ पर इसी सेल में दो न्यूक्लाई में कन्वर्ट हो जाएगा. देन हमारा ये जो एक पेरेंट सेल है वो कैरियोकिनेसिस के बाद साइटोकिनेसिस करके अपने आप को दो डॉटर सेल्स में डिवाइड कर देगा. तो आई एम श्योर कि यू आर गेटिंग this kind of image. So let's begin with the prophase. Most of the biologists and in our book as well, this prophase is divided into three stages. So you can call early prophase, you can call mid prophase, and you can call the late prophase. So the purpose of the prophase, or the jobs that are to be done in this particular phase, they are basically three kinds of the jobs. So the very first job, or the very first task that is to be done in the prophase is called condensing of chromatin into chromosomes. As I said earlier कि before cell division, or if the cell is not dividing, then you will not be able to see any kind of chromosomes in the cell. But as cell begins to divide, or it is going to replicate itself into two daughter cell, so then you will be able to see this chromatin network. This will be converted into the chromosomes. So the very first thing that must be done in the substage called prophase is called the condensing of chromatin into chromosome. The second job is about the division of the centrosome into centriole, and from the centriole the fine thin threads they emerge out of this of the centriole, and they makes a kind of a shape, and that is called spindle shaped. So if you recall, or if you look into the chapter number 4 biology 1, the cell, we have discussed that there are certain microbodies, they are present in the cell of an animal over the nuclei, and they are called the centriole. Remember, these two centrioles collectively are called the centrosome. Their job is to divide the chromosome, but first of all they will arrange chromosomes within itself, then they will divide the chromosomes into the daughter nuclei. Then we will be able to say कि हमने जो है वो क्रोमोसोम्स को डॉटर न्यूक्लाई के अंदर इक्वली डिवाइड कर दिया है, या उनको हमने सेपरेट कर दिया है. सो द सेकंड जॉब, और द सेकंड टास्क ऑफ द प्रोफेज is that we have to form the centrioles, and we have to form the spindle fibers that are made up of microtubules. So we have to form that spindle fibers, or you can say that formation of spindle fibers, and they are emerging out from centriole, the microbodies of the cell. Remember that this is within itself the two bodies, so it will move. This is called a pair of a centriole or a set of centriole. One of this set will move here and other will remain as it is at here, so they will form a kind of a structure which I'll show you later in this process. So this is called the second task of the prophase which is to be done, and this is called the formation of spindle fibers from centrioles. And in these spindle fibers a cell arranges its chromosome for its division in the daughter nuclei. Third task or the third job that is to be done in the prophase is the disappearance nuclear membrane the disappearance of the nuclear membrane and nucleolus on temporary basis. In the prophase, this nuclear membrane which you people are observing and this nucleolus, they will disappear for some time when these chromosomes are divided in the two daughter nuclei. Then at the time of this mitosis phase, when this will end, so these nuclear membrane and nucleolus will reappear again, but they will not reappear in this prophase, they will disappear in this prophase on the temporary basis. So you can call K in the mitosis phase, the first substage is called the prophase, and in the prophase there are three main things that are to be done in this process or in this substage called prophase. The very first thing is called the condensing of chromatin into chromosome. The second is called the formation of spindle fibers from the centrioles, and the third one is called the disappearance of nuclear membrane and nucleolus on its temporary basis. As you people can see as well on the page number 171, this is the diagram of the whole process that you people are called mitosis. So the very first three figures of this page, they are of the prophase. So you people can clearly see that this is called the one centriole, the two bodies, this is another centriole, and at the same time the chromosomes, they are also, or the condensing of the chromatin is also occurring in this region. As I said, the second job or the second task is the movement of this centriole at this position. So you can see the centriole is moving and it is moved in this position. लेकिन जैसे ही ये मूव करना यहाँ से स्टार्ट करता है तो उसी वक्त इसके अंदर मौजूद जो हमारे पास माइक्रोट्यूब्यूल के धागे हैं वो यहाँ से इमर्ज आउट होना स्टार्ट हो जाते हैं. तो एज यू कैन सी क्लियरली कि दिस इज़ कॉल्ड द formation of the spindle fibers. This shape is called spindle. This oval shape, this is called spindle shape, and these fibers are called spindle fibers. I'm sure you will remember The centriole we have discussed in detail, that centrioles are made up of microtubules, the 9 rows of the 3 kind of a filament. So these 3 pictures you can clearly see, this is prophase, this is also prophase, and this is prophase. If you look in this picture, we do not have any kind of chromosomes, but I said कि as prophase starts, condensing of chromatin starts, and by the end of prophase you will not be able to see any kind of chromatin network, but you will be able to see the fine separated threads, what you people are called chromosomes. So this is you can say early prophase, you can call it as mid prophase, and you can call it as the late prophase. You can also look in this picture that we do not have any kind of nuclear membrane. As I said here, the nuclear membrane and the nucleolus, it will also disappear. So here you are not able to see any kind of nuclear membrane or the nucleolus, but you are looking that we have converted all or the network of the what you people were calling chromatin network into fine threads called the chromosomes. So let's discuss once again that if we are in the prophase, so if in the start of prophase we have a network of threads called chromatin network, so by the end of prophase we will have the structure like this, what you people are calling as a chromosome. Keep one thing in mind, that this partner, or you people are calling this as a chromatid, we have made this chromatid in the S phase or in the interphase in the synthesis, which we people were calling as the synthesis of DNA. So हमने जो है इसको finally chromosome में convert कर दिया है. तो this happens in the prophase. और जहाँ तक बात आती है आपके पास इनकी, तो हमारे पास ये जो स्पिंडल फाइबर्स बनते हैं सेंट्रायोल्स में से, ये आपको इस एनिमल सेल के अंदर 3 किस्म के बनते दिखाई देंगे. नाउ यू विल आस्क ए काइंड ऑफ अ क्वेश्चन कि if centrioles are not present in the most plant or in the higher plants, then what happens with them? So basically the cytoskeletal fibers which are present at this region, so they उस न्यूक्लियस में, प्लांट के न्यूक्लियस में, there's a kind of a constriction of those fibers, of those cytoskeletal fibers. तो फिर उनमें जो है वो यही क्रोमोसोम्स अटैच हो जाते हैं जो हमारे पास यहाँ पर एनिमल सेल में स्पिंडल फाइबर में होते हैं. तो इफ़ यू विल लुक इनटू योर बुक एज वेल, तो हमारे पास ये जो 2 सेंट्रायोल्स हैं जो कि अब दोनों ऑपोजिट डायरेक्शन में मूव कर गए हैं, these spindle fibers which will emerge out of this centriole will be of 3 shapes. The very first they will be calling as continuous fibers. It is emerging from this pole and it is going towards this pole. So this will be called as continuous fiber. So this is the one form of the spindle fibers. The second is called discontinuous. This will come to the half of this shape, and you people can also call that equator as well. So this will be called as discontinuous fibers. The third one, they will emerge from this pole, but they will never come to this pole, or they will never come to this half of this shape. So there are 3 kinds of spindle fibers. You can also see in your book as well Here are the three fibers. This is emerging from this pole, and it is coming only to the half of the area. This is kind of a continuous fiber which is going towards this pole, and there are the third which are emerging from this pole, but they are not going to the next pole, or they are not going to the half of its pole. So you can also look this thing here as well. Okay, there are continuous spindle fibers, they're running from pole to pole. There are half or discontinuous spindle fibers, they are running from pole to the equator or the half of the spindle. And the third one, they are called astral fibers. They are short fibers radiating from the centrioles only at one pole. So this is also, this thing which I discussed over here, okay, that the higher plants and some insects lack the centriole. In them, spindle appears. This structure appears, but without this centriole. So from where this structure is appearing in those living organisms, if you remember, the whole cell carries a kind of a network of fibers, what we people called the cytoskeletal fibers. They are present in all over the cell. So the fibers of the cell which are present at the region of the nucleus, they form a kind of a constriction at this area, and the chromosomes of that living organism, they arrange themselves in in that spindle shaped of the living organism. I repeat again, they lack centriole. The higher plants and some insects, they do not possess any kind of these centrioles, but it doesn't mean they do not divide their chromosomes. It doesn't mean that they do not form any this kind of a shape, what you people are called spindle shape. We all know that all living organisms, they possess cell, and if an organism is having a cell, it also have a kind of a network of protein fibers, what we people call them as the cytoskeleton. So their chromosomes, if we have discussed in prophase, we convert this chromatin into chromosomes, and these chromosomes we attach, or a cell attaches their chromosome in this kind of fibers, which, what we are calling spindle fibers. So their chromosomes, the cells, the chromosomes of the plants and the chromosomes of other animals which lack centriole, their chromosomes also divided as the animals divide, but they will not have any kind of centriole. They will only have this kind of a shape. They will have this kind of arrangement in their cell. Now the second phase of the prophase, the mitosis phase, that is called the metaphase. In this phase, there are basically two things that are to be done, but the most important thing in this phase is the chromosomes, what we have seen at the last of the last event or the last time of the prophase. In this metaphase, chromosomes which we converted from, or which we made, they are already made, but we condensed them. So in this phase, chromosomes, they arrange themselves at equator of the spindle fibers. If you recall prophase, so this is called the cell membrane, this is centriole, and we had chromosomes. So in this metaphase, these chromosomes, what we have made from what we have condensed from the chromatin, and we also made this kind of a structure in the prophase. So in this metaphase, these chromosomes will arrange themselves at this, this equator called the half of that of any oval or circle shape. So these chromosomes will arrange themselves in this metaphase at this equator. So the metaphase so this in this phase, the chromosomes, they will arrange themselves at the equator of the spindle fiber. So you people can also call that this is metaphase, and this is the very first job or the very first thing that will happen, that all these chromosomes they arrange themselves at the equator of this spindle fiber. The next thing in the metaphase that will happen is that centromere will also divide. Division of centromere into two. If we look at a chromosome, this is called a mature or a replicated one. This is called the centromere. So in this metaphase, what happens? This centromere divides and this forms in this fashion, but they do not get separate as I have separated them. They are very close to each other. So you can say कि हमारे पास जो एक क्रोमोसोम है, प्लीज़ रिमेंबर दिस इज़ क्रोमोसोम नंबर 1. सो इस क्रोमोसोम को यहाँ पर मेटाफेज के एंड में जो इसका सेंट्रोमेयर है वो डिवाइड होता है 2 सेंट्रोमेयर में, और फिर इस तरीके से हम जो है वो इन 2 क्रोमेटिड्स को आपस में सेपरेट कर देते हैं. अगर आपको दोबारा से याद दिलाऊं तो हमारे पास एक क्रोमोसोम जो है वो 2 क्रोमेटिड्स का बना हुआ होता है, और इन दीज 2 क्रोमेटिड्स we have the same sequence of nucleotides in the DNA. I mean, this possess also a DNA and this also possess a DNA. So if you remember, okay, we had a DNA and we replicated this DNA into 2, और यही 2 DNA हमारे पास इन 2 क्रोमेटिड्स की शक्ल में यहाँ मौजूद हैं, जिनको मेटाफेज के एंड में या मेटाफेज के ख़त्म होते ही हम जो है इसको डिवाइड करते हैं, और ये फिर ऐसे आपके पास 2 क्रोमेटिड्स की शक्ल में यहाँ पर मेटाफेज यानी इस प्लेट पर फॉर्म हो जाते हैं. इफ़ यू कैन सी इन योर बुक, तो इट इज़ ऑल्सो इन द सेम फॉर्म on the page number 171. Look, as I said, these 3 pictures, they belong to the prophase. Now if you look at this picture, so all the chromosomes, they have arranged themselves at the equator, or it is called the metaphase plate as well.